solvent, which moves by osmosis across plasma membrane from region of higher water concentration to lower water concentration, down concentration gradient. During osmosis, water molecules pass through the plasma membrane in two ways. First, by passing through the phospholipid bilayer via simple diffusion. Second, by passing through aquaporins. Osmosis occurs only when the membrane is permeable to water but not to certain solute. Thus, the cell osmosis can be investigated by using the mammalian erythrocyte which contain hemoglobin.
When red blood cells immerse inside a hypotonic solution, water influx is greater than water efflux. This causes the cell permeability increase towards hemoglobin and the cell becomes round and swollen. Hemoglobin and intracellular components diffuse out to outside the red blood cell back to its original shape. In a hypertonic solution, the water entering the cell is lower than water leaving the cell. This causes the red blood cell to become crenulated. In an isotonic solution, rate of water influx is same as water efflux, so the red blood cell maintains its shape.
In this experiment, there are three sections. Section A1, A2 and Section B. In Section A1, the objective is to determine the effect of tonicity, hypotonic, isotonic and hypertonic solution to erythrocyte as the reference to the Section A2 and Section B. In Section A2, the objective is to determine the points of lysis of erythrocyte in different concentrations of NaCl solution. In Section B, the objective is to study the roles of penetrating and non-penetrating solute on the hemolysis of erythrocyte.
Label the tube as such hypertonic, hypotonic, and isotonic. Add 5 ml of NaCl solution with the corresponding concentrations into each tube. Add 5 ml of DDH2O into another tube. Add 50 micrograms of blood into each test tube by using a micropipette. Carefully transfer all the supernatant into clean test tube by using a transfer pipette. Determine the percentage of hemolysis in each tube by measuring the absorbency of each supernatant. Percentage hemolysis is equal to the absorbency of supernatant per absorbency of supernatant from the tube treated with distilled water times with 100. Plot the graph for the percentage of hemolysis versus NaCl concentration.
Today, I would like to present to you the result of the experiment that we get. So before we get into the result, I would like to present to you the percentage of the analysis. So this is the formula of the percentage of the hemolysis. So the percentage of the hemolysis equals to the absorbency of the supernatant over the absorbency of supernatant from the tube treated with the distilled water, the ionized distilled water times 100%. So for section A1, this is the result that we get. So for the deionized distilled water, which we use it as a control, so the result that we get is 2.023, and for hypotonic, the percentage of hemolysis we get is 70.14, and for isotonic is 2.58, and the other one, the hypotonic, is 1.90. So the observation that we get for this four solution is hypotonic is a red solution, which also for the ionized distilled water, and for the isotonic and hypotonic, the result that we get is clear solution with red precipitate, and the red precipitate, which is actually the platelets. Okay, for section A2, this is the result that we get, and the section A2 is to determine the lysis point with against the concentration of the sodium chloride solution. So the observation that we get is the 0.1 from 0.1 to 0.4% of the concentration sodium chloride solution. The observation is red solution, and after the 0.45, the result that we get is clear solution with red precipitate, and the observation is remain until the concentration which is until 1%. Okay, so and for the ionized distilled water, so the result that we get is the observancy is 1.689, which is the observation is also the red solution. So after we transfer this result to the graph, so this is the result that we get. So there is a peak on the 0.4, which is the result that we get is a 101.54, and then after this point, the graph is going down and is going down rapidly, and until about 0.55, the result is maintained here. And for the section B, this is the result that we get, and we are using different type and concentration of the solution. So here, the first one is 300 mOS mole of the sucrose. The absorbency we get is 0.058, and the percentage is 3.43. And for the glucose, which have the same osmolarity, and the result that we get is 0.035, and the percentage of hemolysis is 2.07. And for the sodium chloride, which have the same osmolarity, the result that we get is 0.024, and the absorbency is 1.42. Okay, so the observation that we get for these three type of solution is the solution which is clear and with red precipitate. Okay, then after we use 300 of MOS mole of the urea, and also for 150 and 600 osmolarity for these three different osmolarity of the urea, the result that we get observation is 3 is red solution, but the percentage of the hemolysis for the 300 MOSM mole is the highest, which is 133.21%, and it's going down for the 150, and the percentage we get is 87.33, and for 600, the percentage we get is 67.5. Okay, and then another solution is that we mix the 300 osmolarity with of the urea with the 300 mOS mole of sodium chloride. So the result that we get, the percentage is 3.08, and also which the observation will be clear solution with red precipitate. And the control we use is same with the A2, which is 1.689, and the observation we get is red solution.
Now we will talk about the experiment. So this is about the experiment. From the results obtained, the percentage of hemolysis of hypotonic, hypertonic, and isotonic solutions are 70.14%, 1.90%, and 2.58% respectively. So the 70.14% of hemolysis for the red supernatant shows that some of the red blood cells undergo hemolysis in hypotonic solutions. So when the red blood cells are placed in hypotonic solutions, cells become swollen and round, replacing their original biconcave shapes. This is because the concentration of non-penetrating solutes in hypotonic solutions is much less compared to red blood cells intracellular fluid. There is a net flux in water, thus the water molecules will move down its concentration gradients into the cells. Meanwhile, the cells will increase in cell membrane permeability towards the hemoglobin. The red blood cells will lose their hemoglobin and other intracellular components to the extracellular space. However, there should be no structural destructions of the red blood cells' cell membrane because the cells are capable of to return to their original shapes after losing hemoglobin in hypertonic solutions. So there should be 0.00% of hemolysis. There's the clear supernatant in both hypertonic and isotonic solutions. In the hypertonic solutions, the concentrations of non-penetrating solutes is higher than its concentration in red blood cells intracellular fluids. So the water molecules will move out of red blood cells into the solutions. The cells chelinated and no hemoglobin will diffuse out from the cells. While for the isotonic solutions, the concentration of non-penetrating solutes is equal between the solutions and the red blood cells intracellular fluids. The rate of water moving in and out of the erythrocytes is the same, so there is no net movement of water into the cell. However, from the result we obtained, there is 1.90% of hemolysis for hypotonic solutions and 2.58% of hemolysis for isotonic solutions. So these results obtained are close to 0.00%. The inaccurate results obtained may be due to the human errors while handling the red blood cells. We might accidentally transfer the erythrocytes that contain the hemoglobin, which is located at the bottoms of centrifuge tubes, into the clean small test tubes for measuring of absorbencies of supernatants. We also might have shaken the tube containing the blood hashlete and caused some of the red blood cells to rupture. Other than that, the surface of the plastic container might not be cleaned clearly. So this may increase the readings as the real transmitted out cannot pass through the unclear surface easily, resulting to higher value of absorbency. Now we will discuss about the section A2. So this is the discussions about A2. The graph percentage of hemolysis against the sodium chloride concentrations show that the percentage of hemolysis is inversely proportional to the concentrations of sodium chloride. The higher is the concentrations of sodium chloride, the lower is the percentage of hemolysis of red blood cells. From the graph, the isotonicity point is 0.55% of sodium chloride solutions, where the percentage of hemolysis is close to 0 at this point. At the concentrations of 0.55% sodium chloride, the cell is isotonic to the solutions. The movement of water in and out of the cell across the plasma membrane are at equal rates. Hence, there is no change in the cell volume as no hemolysis takes place. The solutions which lower than 0.55% are hypotonic to the red blood cells. It is found that the hemolysis process have been occurred and the leakage of hemoglobin. So, from 0.45% to 1.0% of sodium chloride, it is found that the clear solutions with red precipitate solutions are formed. There are only a small amount of red blood cells undergoing hemolysis, and the supernatants in the test tubes still appear clearly. And there's only a small amount of hemoglobin that moves out of red blood cells to sodium chloride solutions. For the sodium chloride solutions of concentrations from 0.10% to 0.40%, the color of supernatant becomes red after centrifuge and the percentage of hemolysis is higher relatively. The lower is the concentrations of sodium chloride solutions, the darker is the red color and the greater is the percentage of hemolysis. This is because the lower the concentrations of sodium chloride solutions, the more hypotonic to the solutions to the cell. In hypotonic solutions, there is a net flux and water moves from the solutions into the cell swell and hemolysis occurs. It causes the pores in the membrane become larger and permeable to hemoglobin. Therefore, the solutions in the test tube is red in color due to existence of hemoglobin that contain red pigments. Now, we will talk about the discussions for section B. So, this is the discussions for section B. When the red blood cell is placed in 300 milliosmol of sucrose, the percentage of hemolysis we obtain is 3.43%, which is quite close to zero, and the solution we obtain is a clear solution. This shows that 300 milliosmol of sucrose is isotonic to red blood cells when compared with the results in section A1. The small difference of percentage of hemolysis may be due to the contaminated surface of the plastic container that we are using for taking the readings from the spectrophotometer, causing the inaccuracy of results. Besides, the error could be due to the unclean plastic container which is used to contain the supernatants, and it was used over and over again even after washing. For the 300 milliosmol of glucose, the result shows 2.07% of hemolysis, which is approximate to 0%. Thus, we can conclude that 300 milliosmol is isotonic to the red blood cells when we compare to the results contained in section A1, and also the glucose is a non-penetrating solid, which gives the clear solutions. For the 300 milliosmol of sodium chloride, the percentage of hemolysis that we gain from the experiment is 1.42%. The 300 milliosmol of sodium chloride is isotonic to the red blood cells from the reading we get from section A1. Besides, we can conclude that the sodium chloride is non-penetrating solutes, and thus we can make inference that the concentrations of sodium chloride in red blood cell is 300 milliosmol. For the 300 milliosmol, 150 milliosmol, and 600 milliosmol of urea solutions, the percentage of hemolysis are 133.21%, 87.33%, and 67.50% respectively. The red blood cell in this tube undergoes the hemolysis regardless of the concentrations of urea present. We can then conclude that the urea is penetrating solutes, and the membrane of the cells is permeable to the urea and the water molecules. Where there is a water reflux entering the red blood cell will increase in shape and the volume, and the wall of red blood cell will become enlarged, and thus the hemoglobin will then flow out from the cells, causing the supernatant to be red in color. So below these are one of the example of different concentrations of the urea. For the 300 milliosmol of urea in 300 milliosmol of sodium chloride solutions, the percentage of hemolysis is 3.08%, which is close to 0%. That shows that the solution is isotonic to the red blood cell when compared with the results from A1. In previous experiment, we proved that the sodium chloride is non-penetrating solutes, while urea is penetrating solutes. As a result, the urea tends to move into the intracellular fluid. The mixed solution is now isotonic to the red blood cells as it contains same concentrations of non-penetrating solutes with the cell. In this experiment, we had made 4 conclusions. The first conclusion is that the movement of the water molecules is affected by the tonicity of the solutions and does not depend on the osmolarity of the solution. So the second one will be the percentage of hemolysis is inversely proportional to the percentage of the sodium chloride. And the third one is the sucrose and the sodium chloride are the non-penetrating solutes, whereas the glucose and the urea are the penetrating solutes. Thus, both solutes of sucrose and sodium chloride will influence the hemolysis of red blood cells due to the tonicity of both solutions. And the final one is the erythrocytes will swell in hypotonic solutions, crenate in hypertonic solutions, and no changes of cell volume in the isotonic solution.